Hi, Year 7, and welcome to week 5 of lockdown. I hope everybody is well. As you can tell, we are going to start a section on biology. In fact, we're going to do biology for most of the rest of the year. You've been listening to Mr. Aero's Australian accent, and now you're going to have a chance to listen to my South African accent. I suggest that you click through the PowerPoint as well, either with this video or after you've watched the video, because there's some stuff on there that you may want to see. So, about cells, the building blocks of all living things. I'm not going to give you the answers to these 3 questions. They are for you to think about. When you look at those cells, do you think they are from a plant or an animal? Can you see where the nucleus is? And can you see where the cell walls are? We need to know several things about cells for this lesson. There are 3 of them. We need to know how to describe a cell, how to use a microscope to observe a cell, and how to calculate magnification. We need to know these things really so that next lesson we can look at how we use it and how we prepare slides to look at cells. There's some key words for this lesson on the top right hand side. We need to know what an organism is, a cell, a microscope, how to observe, and how to calculate magnification. So what are cells? Cell comes from a Latin word which means a small room. Here is a small room, also called a cell. And cells are also small rooms because they have a wall around the outside and they contain a variety of things on the inside. So cells are the building blocks for all living things. They're very small, about 10 to 20 micrometers. That small u means micro. That's a millionth of a meter. The smallest can have only one cell, and that's a unicellular organism. And multicellular organisms, like us, have more than one cell. We are made up of about 30 trillion cells. I'm going to leave you to count how many noughts there are. If you're watching the PowerPoint or looking through the PowerPoint, you can click on the cell size animation, which gives you a good idea of how tiny cells really are. On the other hand, here are some unicellular or single-celled organisms. So these pictures are of one cell. The first one is yeast, which is what we use to make bread. And the next one is the largest single-cell organism on Earth. So having told you that cells are really tiny, here's an example of one. It's a single cell that's really big. Generally, they are so tiny we need a microscope to see them. And the microscope was invented by Robert Hooke about 350 years ago, and he was able to observe individual cells of cork. So he looked through the microscope, he saw individual cork cells, and he drew a picture, which we'll see later. So to see objects as tiny as cells, you need a microscope that's powerful enough, you need a dye to make the cells easier to see, and you need a very thin layer of cells so that the light can go through it. I'm quite interested in Robert Hooke, and he's somebody you should know about. It says that he was born in 1635, which is a long time ago, and he was a polymath, which means he was an expert in lots of different fields. So if you look at the green box, you can see that he wrote papers and he studied and he discovered things in all of those different fields of science. Mechanics, gravitation, horology, that means the study of clocks and watches, microscopy, paleontology, which is the study of fossils, astronomy, memory, and architecture. And then you might remember when we were doing forces, we derived Hooke's law, where we put different weights on the end of a spring and we measured the extension of the spring. And that's how we derived Hooke's law. If you look at that picture of Mr. Hooke, just above his head, you can see what's called a water clock. So, as we said, he was interested in horology. And on the shelf behind him, you can see his microscope, the red thing, and you can see a fossil. So that gives you some idea of all the things he was interested in. There's a closer picture of his microscope, and there are the drawings of the cork cells that he observed through his microscope. He even coined the word cell, or he decided to use the word cell to describe. Today we're going to learn about the parts of a microscope. I first of all want to show you the principle on how it works. We've all used a magnifying glass to make the writing a little bit bigger, but if you use two magnifying glasses and you adjust them the right distance apart, you can make the writing even bigger. So that's why microscopes have two lenses. Okay. Now we're going to look at the parts of a microscope from top to bottom. Okay, the top lens, which we used over there, is called the eyepiece. So that's the first lens, and the eyepiece has a magnification of 10 times. So the second lens that I showed you is called the objective. On this microscope there are three objective lenses. There's one with the biggest magnification, one with slightly less magnification, and one with even less magnification. And they have a little click, maybe hear that, when they pop into place. The next thing down is called the stage, and you place your object on the stage, and that's easy to remember because that's where people perform. And you can remember the name of the objective lens because your object is on the stage, and that's the nearest one to the object. So you place your object on the stage and you clip it on with these two clips. Now we have two ways of focusing the microscope. The first one is the coarse adjustment, or the coarse focus. See, as I turn this wheel, the stage moves quite a lot. That's why it's called the coarse adjustment. The next one down here is called the fine adjustment, which is for focusing. And with this one, it's such a fine adjustment, you can probably hardly see the stage moving. Okay, the next part down on the microscope is the light, which shines through, especially if you're doing a biology slide, it shines through it so you can see the cells or the things you're looking at better. And then at the bottom there's a very heavy base. So I'm going to go quickly through the parts in reverse because some people like to learn it that way. So we have a very heavy base, we have the light, we have the fine focus, we have the coarse focus, which is to find your object, we have the stage where you place your object, we have objective lenses nearer the object of different magnifications, and lastly we have the eyepiece lens which you look through. Some people who wear glasses prefer to use their glasses to look through it, and some people prefer to take their glasses off. And that's all I have to say on the parts of a microscope. Oh, I forgot to mention that in some of the microscopes you'll be using, instead of a light at the bottom, there is a mirror which you can adjust to make sure the light is shining through your slide. I hope you enjoyed that little demonstration of all the parts of a microscope. On this slide is a reference point really. I'm not going to go through all of those things because I've done that in the video. It's really just there for you to look at and refer to if you forget what one of the things are. So now I'd like you to pause the video, please. Find your worksheet and label the diagram of the microscope. Once you've done that, on the next slide there is a test. Obviously I've covered up the answers, but the answers will be there, will appear when you click through PowerPoint. Now there is a little video on how to use a microscope, and you need to use this link to go to the video on YouTube. The link is on the PowerPoint itself. If you watch it more than once, you should be able to put all the steps of using a microscope in order. So that's on the worksheet, it's question 2. Watch the video, see if you can put all of the steps in the correct order, where you start to where you finish. The last thing we have to learn about in this lesson is how to calculate magnification. You might remember that I showed you in the very first demo that if you use two magnifying glasses, one above the other, it magnifies more than using just one. And the top magnifying glass we use in the microscope is called the eyepiece, which is near your eye, and the bottom one is called the objective lens, which is near the object. Right. Each lens magnifies the object you're looking at. That big lens that I drew there magnifies it more, and the little lens magnifies it less. But the total magnification of both lenses is the product of the magnification of each lens. Product means multiply. There's a question there that says, are these concave or convex lenses? And I'm going to give you a minute to think about it and then tell you that they are convex. Now this is how we calculate the magnification. Here's an example. So that bigger lens down there on the left has a magnification of 20 times. That's how we say 20x, 20 times. And the smaller lens has a magnification of 5 times. So the total magnification is the product of the magnification of each lens. So in other words, the total magnification is 20 times 5, which is 100 times. Here are some more examples. On the microscopes that you will see in the videos, there are 3 objective lenses, and they each have a different color. We have on the school microscopes a red and a yellow and a blue one. If you look at the picture at the top right, we don't use the white ones from what I remember. So on each of those objective lenses in the picture, it tells you the magnification. The red one is 4 times, the yellow one is 10 times, and the blue one is 40 times. And remember our rule, the total magnification is the product of the magnification of each lens. So, question 1. If the eyepiece always has a magnification of 10 times, what's the total magnification of the red objective lens? So the red objective lens has a 4 times magnification, so we have to do 4 times 10, which is 40 times. So the total magnification will be 40 times. Okay, if the eyepiece has a magnification of 10, what's the total magnification of the white lens? The white lens has a magnification of 100, so the total will be 1,000 times. So question 2. Now the eyepiece has a magnification of 5 times. So what's the total magnification of the blue lens? That will be 5 times 40, which should be 200. And if we want to find the total magnification with the eyepiece of 5 and the yellow lens of 10 times, the total will be 50 times magnification. That's not so difficult. I'd like you to complete the rest of this worksheet. There are some further examples of calculating magnification and a little bit of a research question on electron microscopes if you're interested in finding out about them. They would have used electron microscopes to see what a coronavirus looks like. You'll be pleased to know that this is the last worksheet from this topic. All of the other work is going to be in the form of quizzes inside the PowerPoint. So you've finished the first biology lesson on observing cells, and next week we'll be learning about different types of cell. And I wonder if you can think about, or if you know, what kind of cell there is in that picture. Okay, that completes today's lesson, and I'll see you with lesson 2.